Okay, we are back. I think we're recording. Yeah, we are recording. All right, so we are going to go over the last section that you have to know before the cumulative exam, and that is eukaryotic gene regulation, which I'm going to tell you is really, really complicated, but you don't have to know all of the details. So I will share my screen. Oh, come on. I'm always amazed that this even works, this iPad situation. All righty, so it will be this. Okay, gene regulation. Let's see where we left off. Okay, so we are talking now about eukaryotic gene expression. So remember, eukaryotic cells have membrane-bound organelles. They're larger, they have a nucleus, they have linear chromosomes. So remember all these differences that are going to become important for the final. All right, so we talked a little bit about what it means to turn genes on, right? And what that means is when we turn genes on, we mean the gene is going to be transcribed into mRNA, right? And then eventually made into protein. So when we express a gene or we turn a gene on, that means that RNA polymerase was recruited to the promoter and that RNA was made from that DNA. All right, so just want to give you a quick example of when and why that might be useful. Right, so here's an example of cell signaling, and we haven't talked about cell signaling yet, but we will. So this is just kind of a preview of that. So this is an example of growth factor signaling, and we may have heard, you may have remembered this. This was actually something that was involved in that RAS cancer oncogene as well. All right, so growth factors are chemical signals from a cell that instruct a target cell to grow and divide. All right, so a cell notices this growth factor and decides to divide. So these growth factors came from another cell, and they are going to tell this cell here to grow and divide. All right, so this little yellow ball is the growth factor, all right? But how do these processes actually occur? All right, so you don't have to know all the ins and outs. This is more of a preview for cell signaling, but the outcome you do have to understand. So growth factor binding, so what we're talking about is a receptor on the cell membrane, here in blue, here's the receptor, will bind to the growth factor. Upon binding, we're going to see that the receptor changes shape and triggers a series of chemical events that will activate transcription factors. All right, so here is really cell signaling, the part that you're not really responsible for yet. But what the result is that it activates transcription factors. Transcription factors are something that you should know what that is. Remember, transcription factors will bind to a promoter and act as little flags, being like, hey, hey, RNA polymerase, come here, come hither, right? So it will bind the DNA. Transcription factors will bind DNA promoter, the promoter of a gene, and recruit RNA polymerase, right? So those transcription factors are going to bind the DNA, and they are going to recruit RNA polymerase, which will induce transcription, right? So we're going to see transcription happening. And when I mean transcription, I mean making that RNA, all right? Those RNA are specifically for growth or cell division, right? Those RNAs will go out into the cytoplasm, be translated by the ribosomes, and then we will create proteins that drive growth and cell division. So an example might be cyclin, right? Something like that. All right, so eukaryotic gene expression can be regulated at every single stage, right? Meaning all of them. So that's why it can be really confusing. So here's an example of eukaryotic gene expression, and it can be regulated at every step. So the first one is gene accessibility. So for example, how tightly wound around those histones is the DNA? If it's super tight, it's not very accessible for RNA polymerase, but if it's looser, it is. The second one is at the transcriptional level, right? So you can have gene regulation at the transcriptional level. The third is at RNA processing, right? So remember, you can have all of that alternative splicing to create lots of different kinds of mRNAs. The fourth level is at the mRNA stability. So how stable is that mRNA in the cytoplasm? Is it going to be degraded by enzymes immediately, or is it just going to be stable for a longer period? The fifth is at translation, so the ribosomal work. And then sixth, at that active protein level. So eukaryotic gene expression, remember, has lots of different layers because it's a lot more, these cells are a lot more complex. So therefore, gene regulation is much more complex. All right, so again, these regulation can occur at all of these different levels: DNA accessibility, transcription, RNA processing, RNA stability, translation, and protein activity. I'm not going to get crazy about you learning every single detail about this, but you should know some basics. All right. So, for example, for DNA accessibility, genes exist in these highly compacted chromatin, meaning with histones, right? And those histones can be modified to see whether they're going to be looser or more tightly wound, right? I'm not going to make you know all of the modifications like acetylation or methylation. But for example, if I showed you a figure of histones that have been modified that either loosen the DNA or tighten the DNA, you should know that tighter means less accessibility, less transcription, all right? The second one here is at the transcriptional level, right? And so this is one thing you do have to know, so I'm going to highlight it here. You have to know this new term called enhancers. So remember how prokaryotic DNA had operators where proteins bound and kind of regulated operons. Enhancers are in eukaryotic DNA. They're short regions of DNA that can be bound by activators to increase the likelihood of transcription. And these enhancers can exist very, very far away from the gene of interest, actually like thousands and thousands and thousands of base pairs away. So you can get this DNA looping, right? So you can get gene regulation, transcriptional regulation from really far away from your gene, right? So you can have enhancers are pieces of DNA where proteins can bind and therefore activate transcription, right? The third one is RNA processing. And again, this just means alternative splicing. So different ways that your introns can be sliced. So from a single mRNA, you can make different proteins depending on which introns are spliced out, right? 4 is RNA stability. So every single mRNA has a specific lifespan, right? 5, translation. So initiation of translation can be controlled. We're not going to talk too much about that. And then 6 is that proteins also have kind of a half-life, meaning proteins can also be degraded in the cytoplasm. And they do it through this cool organelle called the proteasome, which is like the garbage disposal. Proteins go out and all those amino acids can be recycled. All right, so very similar to what we saw in prokaryotic cells, we have transcriptional activators, which will increase the likelihood of transcription, right? And we have repressors, which will decrease the likelihood of transcription, right? And these activate the And we also have, as we learned about, transcription factors that can bind, right? And the big difference between prokaryotic and eukaryotic transcription factors is that the eukaryotic ones can bind very, very far from our gene, up to 1 million bases away, right? So binding sites for transcription factors can be very close to the promoter or very far from the promoter. And so, for example, this, I'll highlight it in red, this piece of DNA would be the enhancer, right? Because a protein is binding to that, and we're going to see an increase in transcription. So the main difference between prokaryotic and eukaryotic, again, eukaryotic, this regulation can occur from very, very far away. All right, so let's give a scenario here. Suppose a gene needs to be turned on in your spine, skull, and fingertips, but not the rest of your body. So how can transcription factors make that happen, right? So here are our tissue-specific enhancers, right? And the enhancer, again, is a region on DNA where transcription factors will bind to increase the likelihood of transcription, all right? So for example, here is our gene, our TBX4 gene, and we have enhancers that again are all over that chromosome. We have one here in front of it for the hind leg, we have other ones down here, right? Because eukaryotic regulation can happen from far away. So what happens is that the proteins that bind to those regulatory DNA or those enhancers are tissue specific. So for example, we have an activator protein in, what did we say? Spine, skull, fingers, right? And we might have a repressor protein everywhere else, right? So the proteins that bind to these enhancers can themselves be tissue specific, expressed in only certain tissues, right? And the other very important thing about eukaryotic gene regulation is that it's combinatorial, all right? And you do not have to know all of the details about this, but it's important to know that many genes are controlled by several different transcription factors with a specific combination needed to turn them on. So for example, and these images are directly from the Khan Academy. If you want to go look at their article, it's great. So for example, here's our gene. So this gene is only expressed if both activators are present and the repressor is absent, right? So here our binding sites are kind of like enhancers, right? And then these would be our transcription factors, some activators and some repressors. We'll just call them repressors and activators. I think that's simpler. So we only get transcription when we get both activators bound, right? If we have only one activator bound, we get very little, but some transcription. If we have the repressor bound, we get no transcription. So eukaryotic gene regulation at the transcriptional level is just really complicated. It can occur from very far away, and it can involve several different proteins binding to that DNA. All right, so splicing. So we've already talked a little bit about alternative splicing. And that is the concept where you can have a single pre-mRNA, and then you can get several different mature mRNAs out of that, depending on which introns get spliced out and how that gets spliced out. So in this mRNA, we have spliced out intron 2 all the way through intron 3. So we've gotten rid of that whole shebang. Oops, oh man, I think I did that wrong, sorry. So in number 1, we got rid of that intron and all the way through those, right? So we got 1, 2, 4. In this protein, mature mRNA, we got rid of exon 2, right? So you result in different mature mRNAs. So by definition, alternative splicing is that different cell types may express different regulatory proteins, so that's not really the best definition. So this is the best. Different exon combinations can be used in each cell type, leading to different proteins, right? So a single pre-mRNA can, oh my gosh, yeah, give rise to many different mature mRNA sequences. All right. So regulation of translation or proteins can occur through our favorite phosphorylation, right? And remember, phosphorylation is when you're adding a phosphate group to a protein, right? Adding a phosphate group can act as a molecular switch, right? So it can either turn on a protein or turn off a protein. And just as an example, and this is the kind of thing, here, I'll highlight right here. This is the kind of diagram that I would show you that you've never seen before, that you should be able to interpret what is happening, right? So in the top, we have phosphorylated, this EIF2, and we get no translation happening, all right? However, you should notice that when EIF2 is dephosphorylated or not phosphorylated, then you get an increase in translation, right? This is just one example of how phosphorylation of proteins can change how the ribosome functions, right? And again, I'm not going to go crazy with the details, but I would show you an example of something you've never seen, and you need to interpret what's happening in that figure. All right. All right. And the last but not least is the regulation of proteins. And mostly I'm telling you about the proteasome because it's such a cool organelle, right? So the proteasome is the cell's garbage disposal. Proteins go in, single amino acids come out, right? So we are putting a protein in, it gets degraded, and these amino acids can then be recycled, right? And the way that that happens is that proteins get tagged for disposal, and it gets tagged through this cool little protein called ubiquitin. And guess why it's called ubiquitin? Because it's ubiquitous. Every cell that we have looked at least in eukaryotic cells, I don't know about prokaryotic cells. I'll have to check on that. All eukaryotic cells have ubiquitin. They're little tags that if you get a bunch of these built up on a protein, that means you are going to the garbage disposal, the recycling center. All right, so that's it for eukaryotic gene expression. Key takeaway points. It happens at every single level of that whole through transcription and translation. At the transcriptional level, DNA regulatory regions called enhancers can exist very far from the gene. And it's a lot more complicated than prokaryotic cells. All right, chip, chip, cheerio. Remember, there are questions at the end of these PowerPoints that you should quiz yourself on, and come to office hours and we can chat about them. Mwah. Bye. Adios. Cheers. Peace. Toodles, poodles.